Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, with our ongoing series on development biology, today we would be talking on early development in amphibians, and for this very discussion we have once again with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. So let's welcome our guest Dr. Charu Dogra Rawat and let's start with our lecture on early development in amphibians. Hello ma'am, welcome to the lecture.
Good afternoon everyone. So today we will be talking about the early development in amphibians, and we will be doing it up to gastrulation. So we will just overview take how fertilization happens, what are the cleavage, going from a single cell to a multicellular organism, and then how does the gastrulation occur. So we will do that in detail, that how this multicellular ball of cells get into the multi-layered body plan of the adult. So, the specific objectives for today's session would be to understand the fertilization, cleavage and precondition for gastrulation in amphibians, and we will also understand the fate map and study various techniques of fate mapping. So, looking at the in amphibians, the fertilization occurs anywhere in the animal hemisphere of the amphibian egg. So, amphibian egg is a mesolecithal, telolecithal egg. And it has one area which is a little yolk free, which is called as the animal pole, and that is called as the animal hemisphere, and the lower portion has lot of yolk deposition, and it is called as the vegetal hemisphere. So we have already talked about this bipolar differentiation, and so sperm can practically enter anywhere but on the animal hemisphere of the mature egg. The point of sperm entry marks the future dorsal side, future ventral side of the embryo, and the site 180 degrees opposite to the point of sperm entry marks the dorsal side. So actually the sperm entry determines the dorsoventral axis. We also talked about in general that there has to be three axes that need to be determined: the anterior posterior axis, the dorsoventral axis, and the left and the right axis. While during development. So the dorsoventral axis is the determination is actually right at the time of fertilization in amphibians. So the point of entry becomes the future ventral side, and the 180 degree opposite to it becomes the future dorsal side of the embryo. The sperm centriole, after entering into the egg, organizes the microtubules of the egg and causes them to arrange themselves in a parallel array in the vegetal cytoplasm, separating the cortical cytoplasm from the yolky internal cytoplasm. So the sperm centriole, which enters the egg, kinds of produce these or organizes the microtubules of the egg in a manner so that there is a difference in the organization of the cortical or the peripheral cytoplasm and the internal cytoplasm, which is yolk laden. These microtubular tracts allow the cortical cytoplasm to rotate about 30 degrees with respect to the inner cytoplasm, and this is called as the cortical rotation. So after the fertilization, the event of cortical rotation happens or cortical reaction happens that exposes a region which is called as the grey crescent area. This rotation exposes a band of inner grey cytoplasm in the marginal region or equatorial region of the one cell embryo directly opposite to the point of sperm entry, and this region, the grey crescent region, is where the gastrulation begins. So the figure clearly reveals that this is the egg, and you can see that this is the peripheral cytoplasm and this is the inner cytoplasm. So if this is the point of entry, then this cortical cytoplasm with respect to the inner cytoplasm will rotate around 30 degrees of angle, and during this rotation it will expose this grey crescent area on the opposite side to from where the sperm entered, and this is done basically by this microtubular arrangement done by the sperm centriole which is there, and this grey crescent area marks the future dorsal side of the embryo from where the gastrulation will start. So this is very important because the dorsoventral axis is determined by the point of sperm entry and the appearance of this grey crescent area that marks the future dorsal side. So, looking at reviewing the cleavage in amphibians, it is an unequal radial holoblastic cleavage. So, you can see the first cleavage begins at the animal pole. So, as we have already talked about, the animal pole is relatively yolk free and the vegetal pole is highly yolk laden. So, and cleavage furrows are inhibited by the accumulation of yolk. So, more the yolk, the cleavage is inhibited. But it is a holoblastic cleavage. The cleavage divides the entire egg into blastomeres. Therefore, the cleavage happens, but it is in a different manner. It is until, like for example, until the first division begins and it is not yet completed than the second division. So, the rate of cleavage furrow passing through the animal pole and passing through the vegetal pole is basically differed. So, it becomes slower when passing through the vegetal pole. So, the first cleavage division in amphibians begins at the animal pole and slowly extends down the vegetal region. While the first cleavage furrow is still cleaving the yolk cytoplasm, the second cleavage has already started near the animal pole, and this cleavage is at right angles to the first one and is also meridional. So, passing vertically and through the center of the egg. So, this is meridional and this is again the perpendicular to the first cleavage. The third cleavage, as expected, is equatorial. So, it is equatorial, but however, there is a vegetal cytoplasm, it is a little displaced towards upper side and it is said to be latitudinal. So, you can see that now it is dividing the egg into unequal parts. So, this third division is actually dividing into two kinds of blastomeres. The one are the little ones on the towards the in the animal hemisphere are termed as the micromeres, and the later one are termed as the macromeres. So, this will keep on happening, and this you can see the third cleavage divides the frog embryo into 4 small micromeres and large vegetal region blastomeres, which are called as the macromeres. So, they are highly yolk laden cells. As cleavage progresses, the animal region becomes packed with numerous small cells, while the vegetal region contains only a relatively small number of large yolk laden macromeres. So, micromeres and macromeres, the number of micromeres is much more than the number of the macromeres which are found in the vegetal region. An amphibian embryo containing 16 to 64 blastomeres is termed as a morula. Then after this 64 cell division at the 128 cell stage, the blastocoel becomes, so there is a cavity that appears and this cavity becomes apparent, and this kind of at this stage the frog embryo is called as the blastula. So, this blastocoel or this cavity is very important, and in fact they it has been found that this blastocoel does not initiate at the 128 cell stage, but in fact during the first cleavage also, when the cell is dividing into 2 blastomeres, then also there is a little difference between the attachment of the 2 blastomere that creeps in. So, blastocoel practically starts forming maybe right at the initiation of the cleavage. So, what is the significance of this cavity? So, significance of this cavity, the blastocoel probably serves 2 major functions. First is it permits cell migration during gastrulation. So, gastrulation we have talked about. The general mechanism of gastrulation is basically the attainment of a multi-layered body plan. So, multi-layered body plan, mammals for example are triploblastic, they have got an ectoderm, an endoderm and a mesoderm. And then tissues and organs are derivatives of these 3 germ layers, or 1 or 2 germ layers they can also be derived from. So, these 3 germ layers, the 3-layered body plan has to be achieved from a single ball of cells. So, in the single ball of cells, the blastula, the cells are random, they are lying random, and now they have to migrate to attain their position, that usually the ectoderm is on the outside, the mesoderm is in the middle layer and the endoderm is the innermost layer. So, to attain their positions they need to move, and for the movement they need space. So, the movement, the space that is needed is basically permitted due to the presence of this blastocoel, so that the cells can actually move along and attain their final positions. Then another important significance of blastocele is that it prevents the cells beneath it from interacting prematurely with the cells above it. So, as we will see the gastrulation part, and we will understand that the cells in the animal pole and the cells in the vegetal region, they have different fates. They are going to become different germ layers. For example, the superficial layers are going to become, superficial animal cell layers are going to become the ectoderm. However, the vegetal layers are going to become the endodermal lining. So, the fates are different. And then they also have a mechanism which is called as induction, where one cell influences the fate of the other cell. So, right at the initiation, the cells which have different fates are kind of separated, so that they cannot induce each other prematurely to become something else. So, that is also one of the significance of the blastocele, that this cavity, why does it appear? So, at 128 cell stage, when this blastocele appears, then this is termed as a blastula, and this blastula is a coeloblastula. We have already talked about the types of blastula. This is a coeloblastula because it does contain the coelomic cavity which is there. Then it is also called as an amphiblastula because generally it contains two different types of structures, which is the micromeres and the macromeres. So, amphibians have a coeloblastula and a amphiblastula which is formed. Many cell adhesion molecules keep the cleaving blastomeres together. So, how are the blastomeres tightly packed? They are tightly packed, and this kind of a cavity is created because of certain molecules such as epic adherin. So, epic adherin is provided in the egg cytoplasm. So, the amphibian genome does not produce it. It is already, the mRNA is already present in the egg, and this epic adherin makes the micromeres to adhere together. If there is a mutation in this epic adherin, then there is no prominent blastocele that is visible, because all the cells become loose and they are not tightly packed, and there is no appearance of the promiscuous blastocele which is shown in the blastula stage which is there. So, such molecules tend to adhere the blastomeres together. Now, the next aspect is that after the cleavage, when the multilayered body plan has been occurred, then the embryo has to prepare for the gastrulation. So, the three germinal layers have to be formed. The precondition for the gastrulation to occur is the activation of the genome. So, particularly in frogs, particularly in amphibians, the activation of genome does not occur until the late 12th stage. So, initially we have also talked about that the cleavage divisions are synchronous, and this synchronity is because it is only dependent, all the blastomeres are dependent on whatever mRNA or the proteins are already accumulated in the egg cytoplasm. So, they do not manufacture their own proteins or their own mRNA, or their genomes are inactivated. So, after the 12th cleavage, then the activation of the genome starts, and the synchronity is lost because in different blastomeres different genes can be transcribed. So, in xenopus, until late in the 12th cell cycle, nuclear genes are not activated. At that time, different genes begin to be transcribed in different cells, and the blastomeres acquire the capability to be motile. So, they now have to migrate to their proper positions. So, the blastomeres acquire the capability to be motile. This dramatic change is called as the mid-blastular transition. So, MBT or mid-blastular transition is really important for setting up the stage for the next event, which is the gastrulation. Now, demethylation of certain promoters play an important role in regulating the gene expression at MBT. So, we are talking about that in MBT the gene expression is activated. So, in frog, particularly in xenopus, both the paternal and maternal chromosomes have promoters which are hypermethylated. So, this methylation actually inactivates them. So, demethylation of certain promoters, they tend to regulate the gene expression. After the chromatin is remodeled at the promoters, various transcription factors could bind to these promoters and initiate transcription. So, transcription factors such as Veg-T, now they were already present, or mRNA of these transcription factors are already present in the maternal cytoplasm, but when the chromatin remodeling of the promoters takes place, the promoters becomes demethylated, they become ready to bind transcription factors. So, these transcription factors which are also present in the egg cytoplasm can now bind to these promoters and initiate transcription, and the genome is activated, and then the cell specific proteins are produced. So, to start for this motility and to start for this movement, this kind of activation of the genome is a precondition that is required. Now, we will follow up the amphibian gastrulation, but to get into the detailed mechanism, we will first have to look at and find out that what cell becomes what in the 3 germ layers. So, we will have to follow the fate of the cells which is there. So, different species gastrulate in different ways to achieve the same goal of bringing inside the embryo the areas of the blastulae destined to form the endodermal organs, surrounding the embryo with cells capable of forming the ectoderm, and placing the mesodermal cells in proper positions between them. So, the formation of the 3 germ layers, the ectoderm on the outside, the endoderm on the inside, and the mesodermal layer in between, that is the aim of the gastrulation. However, different species of amphibians there are many ways in which they can gastrulate, but the most studied and studied gastrulation is in xenopus, and therefore, we will follow of the xenopus. Now, the different cells of the blastulae have different fates. So, we are talking about that blastula is a random ball of cells. Now, every cell has a different fate, or each cell has different fate. So, we will have to look at, before starting with the gastrulation, that how it happens and what is the detailed mechanism. We have to look at that what are the fates of these individual cells, or basically the regions, where they have to go and whether they have to become the endoderm, the mesoderm, or the ectoderm. So, for that we have to define what is called as a fate map. So, the term cell lineage is basically following the individual cells to see what they become. So, cell lineage is followed and a fate map is drawn. For example, it is a diagram that map the larval or adult structures onto the region of the embryo from which it arose. So, these are the 4 examples which are given here. And to mention again that any diagrams which are not referred in here are taken from the Gilbert, and as Gilbert is recommended as a textbook, and therefore everything figures and everything reference is basically taken from Gilbert developmental biology book until and unless they are referred. So, these are the 4 fate maps of zebrafish, frog, mouse and chick which are shown here. So, for example, look at the amphibians. So, different colors mark different layers, germ layers. The yellow will give rise to the endoderm. So, most vegetal portion is basically giving rise to the endoderm. Then superficial layers of the animal hemisphere and the marginal zone, we will talk about it more. So, this dark blue is giving rise to the neural ectoderm, and this region is giving rise to the surface ectoderm or epidermal ectoderm. So, epidermal ectoderm, neural ectoderm, and the inner layers, this deep layers are basically giving rise to the mesodermal tissue, and in that mesodermal tissue the central portion will give rise to the notochord. So, that is the kind of an egg. It is just showing that this is a rounded egg and these layers have to form in this manner. So, the gastrulation has to occur so that this yellow portion goes inside, because they are the endodermal cell. The red lies in between because that is the mesodermal tissue, and followed by the blue which is surrounding the egg. So, that is the kind of arrangement. So, and this kind of a diagram is termed as the fate map which is there. So, before talking about the amphibians, right here we will talk about different techniques to generate different fate maps. They can be generated in different ways. One is by observing living embryos. Embryos of certain invertebrates are transparent, have relatively few cells, and the daughter cells remain close to one another. So, it is just possible to look through the microscope and trace the descendants of a particular cell into the organs they generate. Edwin G. Conklin studied eggs of the tunicate Styla patata and followed the fates of every cell in the embryo until each differentiated into particular structures. Styla eggs are peculiar in that the different cells contain different pigments. So, it's easier to follow cell fate, or it is easier to follow cell lineage, because just by observing and looking at individual cells and where they are going and where they are forming, and in fact in Styla they were differently colored also. So, it's much easier, and scientists can generate a fate map just by looking at the embryo only. Other technique involves the vital dye marking. So, trace the fates of different areas of amphibian eggs by applying vital dyes to the region of interest. Now, vital dyes only stain live cells, but they do not kill them. So, they do and stain the cells, but they do not kill them. So, vital dyes, the dyes were mixed with agar and spread on a microscopic slide to dry. The ends of the dyed agar were very thin. Then chips were cut from these ends and placed onto the frog embryo. So, this is the frog embryo which is growing. So, these chips were taken from the agar which contained the vital dye, and they were placed at different regions. Then after the dye stained the cells, the agar chip was removed and cell movements within the embryo could be followed. So, the cells were not dead. So, this chip was placed over the cells, the stain went into the cells were still live, they will still follow their movement, and we can also follow the movement because now they are stained and we can trace that where these cells are going. For example, in this case they have placed it here, and now they can follow that, how they are becoming the part of the neurula, and this might be the neural ectoderm which is there. So, this is how the fate map can be generated. Other methods include the radioactive labeling and the fluorescent dyes. A donor embryo is usually grown in a solution containing radioactive thymidine that becomes incorporated into the DNA of the dividing embryo. Host embryo is grown under normal conditions. The region of interest is cut from the host embryo and is replaced by graft from the donor embryo. After a while, host embryo is sectioned for microscopy. Cells that are radioactive are descendants of the graft and can be distinguished by autoradiography. So, in this case the transplantation will take place. The donor is basically given a radioactive dose, and the cells are grown in a radioactive solution, they incorporate radioactivity, and then a piece is taken and grafted onto the normal embryo. So, it will grow as the normal embryo is growing, but it will have radioactivity that can be detected by the autoradiography, and thus we can follow the cell lineage and draw a fate map. So, that is how the radioactive labeling occurs. But then vital dyes and radioactive markers, they usually have a shelf life. After a time, with each cell division, then each become difficult to detect because the dyes are diluted with each cell division, and it becomes obscure and less receptive to for visibility. So, the fluorescent dyes can be used. They are so intense that once injected into individual cells, they can still be detected in the progeny of these cells many division later. So, they follow the same method as the radioactive labeling or the vital dye staining, but the dyes have a very long shelf life, and therefore, they can be detected for even in a very unstained background, even the smallest amount can be detected. Then another mechanism of generating a fate map is by the genetic marking. One permanent way of marking cells and following their fates is to create mosaic embryos in which the same organism contains cells with different genetic constitutions. For example, chimeric embryos are generated consisting of a graft of quail cells inside a chick embryo. Genetic marking is basically when you create mosaic embryos in which two different structures derived from, or two different tissues derived from the different organisms are put into one. So, same organism contains cells with different genetic composition. So, the quail and the chick which we are talking about, these are two very similar developmental following organisms, but their two genetic composition is different. So, the chick and quails develop in a very similar manner, and the grafted quail cells become integrated into the chick embryo and participate in the construction of various organs. Quail nuclei can be differed from chick nuclei easily, as quail nucleus has very much condensed DNA which is concentrated around nucleoli. Cell specific antigens that are quail specific can be used to identify individual quail cells even if they are hidden within a large population of chick cells. So, creating mosaic organisms where one is genetically different from the other and then tracing and following the cell lineage of the tissues arising from the different genetic composition organism can also generate fate maps, and there is no problem of the dilution or anything, and some specific antigens can also be used to detect the different genetic composition tissues against the background of a large population of the homogeneous genetic background tissues. So, these were some of the techniques and some of the ways by which the fate map can be generated. So, before we start with the gastrulation, we can have a look at the fate map of the Xenopus frog, so that we understand that which regions are going to form the part of which germinal layer. So, this is the fate map of the blastula of the frog of Xenopus laevis. As you can see, this is from exterior. So, exterior we will in fact talk about two different type of cells, the superficial and the deep cells. So, in outside surface you can see that basically there are only two types of the cells composing the outside of the blastula is basically only giving rise to the ectoderm. This is giving rise to the epidermal ectoderm, and this is giving rise to the neural plate ectoderm, and then animal hemisphere cells, the vegetal hemisphere cells, and this is basically termed as the marginal zone. So, animal hemisphere cells, you can see that there is an endoderm. So, this is a subblastoporal endoderm and this is a suprablastoporal endoderm. So, the animal hemisphere cells on the exterior are ectodermal, will give rise to the ectodermal germ layer, and these are the endodermal layer, and here you can see the blastopore is developing here, and that is why it is called as the suprablastoporal and subblastoporal endoderm. So, blastopore is the point from where the gastrulation starts. So, the blastopore pore it mentions as a hole. So, all the cells which has to go inside goes through this blastopore which is there. So, this is a section of the blastula, and in the interior you can actually see that this was the neural plate ectoderm, this was the epidermal ectoderm, and this was the endodermal region, and particularly in the marginal zone, the inner cells which are there is going to give rise to the mesoderm. The ones near the blastopore, the one which is here in the front, will give rise to the somites and the notochord. It is called as the chorda mesoderm. So, this is giving rise to the chorda mesoderm, and in fact this central entire marginal zone cells are giving rise to the somites. The heart or the anterior blood islands or other kind of organs will develop from this portion which is right in the middle, somewhere towards middle of the inner blastula which is there, and this ventral portion will give rise to, this is the lateral plate mesoderm which will give rise to the different organs. So, you can see that the mesoderm will generally originate from the deep layer cells which is lying in the marginal zone. In the animal hemisphere, the exterior cells will give rise to the ectoderm, and in the vegetal hemisphere, the endodermal cells are the ones which are giving rise to the endoderm which is there. So, different cells have different fates depending on whether they are located in the deep or superficial layers of the embryo. The mesodermal precursors exist mostly in the deep layer of the cells, while the ectoderm and the endoderm arise from the superficial layers. So, this is how what the fate map looks like, and then we have to follow the gastrulation of these individual layers. The precursors for the notochord and other mesodermal tissues are located beneath the surface in the equatorial region of the embryo. So, mesodermal precursors, basically this is the chorda mesoderm, they are lying in this marginal zone beneath the surface in the equatorial region which is there. So, we will follow up the detailed mechanism of gastrulation in Xenopus, and just reviewing the fate map of the frog embryo. So, we can see that the superficial layers in the animal hemisphere are going to give rise to the ectodermal germ layer. You know, these are the cells of the ectoderm. This region is the region of the epidermal ectoderm, which are going to form the surface of the embryo. And this region is going to be the neural plate ectoderm, which is going to form the neural tube, you know, the brain and the neural tube. And the lower vegetal hemisphere cells are going to give rise to the endodermal regions, which is basically the lining of the gut. And this is the blastopere region. So, the mesoderm develops from the deep cells which are present in the equatorial region. So, you can see that the one near this blastopore where it is developing, this layer is going to give rise to the somites and the notochord. So, this is basically the entire notochordal or chorda mesoderm it is termed as. Then this region is going to give rise to the heart, anterior blood islands, you know, other mesodermal organs, derivatives. And then this is what is going to give rise to the lateral plate mesoderm. So, mesoderm usually develops from the deeper layers of the blastula, and the surface layers or the superficial layers in the animal hemisphere give rise to the ectoderm, and the vegetal hemisphere give rise to the endoderm. So, let us follow the gastrulation, how it occurs. Looking at the early gastrulation, we will follow up the vegetal rotation and the invagination of the bottle cells. The gastrulation in frog embryo initiated at the future dorsal side of the embryo just below the equator in the gray crescent region. So, we already talked about the gray crescent region, that you know there was a cortical rotation after the egg was fertilized, and this cortical region exposed gray crescent area which was supposed to be the future dorsal side. So, the gastrulation in frog is initiated at this future. So, sperm entry was somewhere from here. This is the animal pole, this is the vegetal pole, and this is the part, and you know it is already mentioning this is the blastocele. These are the superficial layers and these are the deeper layers. So, we already know that the superficial layers have a different fate than the deeper layers, and this is termed as the marginal zone which is there. So, the gastrulation actually starts at this dorsal blastopore or the dorsal side, future dorsal side of the embryo just below the equator in this gray crescent region which is there. The cells invaginate to form a slit like blastopore. So, you can see that the cells have invaginated and they form a slit like blastopore or an opening which is there, and this is formed because the shape of the cells change dramatically. The main body of the cells is displaced towards the inside of the embryo while maintaining connection with the outside surface by a slender neck. These are termed as the bottle cells and they line the archentron as it forms. So, the first cells to start the gastrulation or invagination are these bunch of bottle cells which are there, and their shape changes, and that is why they are termed as bottle cells, because they have a base which is wider, and this base is which are the cells lying here with a slender neck which is outside, and because of this slender neck, you know, the space is created and it becomes a slit like blastopore. So, that is what is happening in here. So, how does it originate? The internalization of the endoderm and the mesoderm is initiated by a movement which is called as vegetal rotation. And in fact, 2 hours before the bottle cells are formed, the internal cell rearrangement propel the cells of the dorsal floor of the blastocele towards the animal cap. So, now you can see in here, this is, you know, it is shown on the left side now, but this is basically the prospective ectoderm. This is the region, the marginal zone region, which is going to give rise to the mesoderm. So, they have differentiated the mesoderm into chorda mesoderm and het mesoderm, and IMZ refers to as the involuting marginal zone. So, involuting marginal zone is basically giving rise to the mesodermal region which is there. So, this is the site of the blastopore formation. This is the vegetal endoderm, which is basically the vegetal cells which are lying, and on the top of it there is the region of pharyngeal endoderm which is there. So, as you can see here, even when the bottle cells are not formed, there is a kind of rotation that has occurred in here. So, this vegetal rotation that has occurred is actually pushing this pharyngeal endoderm towards the roof of the prospective ectoderm which is there. So this rotation, this vegetal rotation places the prospective pharyngeal endoderm adjacent to the blastocele and immediately above the involuting mesoderm or involuting marginal zone. So this cauda mesoderm and head mesoderm, it was not lying above and in front of it, it was lying on a side of it, but because of this vegetal rotation, it pushes this pharyngeal endoderm and now it is lying on the in front of or on top of this involuting marginal zone. These cells then migrate under the basal surface of the blastocele roof. So this is the blastocele roof and now it will migrate from here and it will go on. So the first cells, although it is by a passive movement, it is because of the movement, the vegetal rotation that has occurred. So because of the passive movement, the first cells to go inside and is basically the pharyngeal endoderm which is there. The superficial layer of marginal cells is pulled inward to form the endodermal lining of the archentron. So this is the superficial layer of the endodermal cells. So now they are pushed inside. Now you can see the invagination is occurring. This is the blastopore which is there. So the invagination has started occurring. So because of this invagination or the bottle cells are formed, the superficial layers of are pushed inside, are pulled inside, and they become the endodermal lining of the gut cavity or the archentron merely because it is attached to the actively migrating deep cells. So as I said, the first movement is basically a passive movement. The deeper cells are moving because of this vegetal rotation that has occurred, and because of this active movement of the deeper cells, the superficial cells are just pulled inward. So they are just pulled inward passively. So in this you can again see that the vegetal rotation is occurring and now pharyngeal endoderm is moving at the basal surface of the blastocele, and now here there is a kind of an opening, the proper invagination has occurred, and this invagination has now created, you can see the yellow cells, endodermal cells in here, and there is again a line of yellow endodermal cell here. So this cavity which is now developing is going to be the primitive gut or the archentron cavity. So the bottle cells are the one which are following, taking, they always move towards the anterior side, anterior or the ventral side. So they are going inside, and this cavity is called as a primitive gut cavity, and now you can see it is lined by the endodermal, although this endoderm has moved in passively because of this vegetal rotation or deeper layer cells which have actively moved in. So vegetal endoderm movements push the pharyngeal endoderm forward, driving the endoderm passively into the embryo and towards the animal pole. And this also reflects another important phenomena, that as the cells are moving, the composition of the blastopore will keep on changing. Right now it is basically formed by the endodermal cells, but we will see as the these cells start moving inside, the mesodermal cells start moving inside, they will form the cells of the blastopore. So the composition of the cells of the blastopore will keep on changing. So what happens during the mid-gastrulation region is the involution that occurs at the blastopore lip. So invagination is a gastrulation movement which basically give rise to a pit. When invagination, when a single layer of cells just deepens and form a kind of a pit. Involution is when the cells start moving inside but start growing over or above or below another layer of cells which is lying there. So that kind of gastrulation movement is basically called as involution. So the next phase involves the involution of the marginal zone cells, while the animal cells undergo epiboly and converge to the blastopore. So we will follow individually this part. So first we will look at how this involution of the marginal zone occurs. So you can see here the blastopore started developing here, and the first layer was the bottle cells which were going inside, which are now forming the roof of the archentron. So they have reached up till here. This is the developing archentron which is there. It is lined on both sides with the endodermal cells, and this red cells have also starting involuting. Now they start growing below this blue layer, and therefore it is termed as involution. So first it starts forming, this is the dorsal lip of blastopore, then it starts happening laterally, and then it also starts happening ventrally, which is on this side. So, this is a section of the blastula and the blastocele is actually displaced. So, when the migrating marginal zone cells reach and become the dorsal lip of the blastopore, they turn inward and travel along the inner surface of our outer animal hemisphere cells. So, you can see this is the marginal zone cells. In marginal zone cells, you have got these superficial cells which will give rise to the endodermal region, and you have got these inner cells which will give rise to the mesodermal region. So, this marginal zone cells, when they will start, this is how the blastopore is formed. So, blastopore is formed just here. These are the bottle cells, the dramatic change, shape change. So, this is basically the blastopore which is here, and because of this vegetal rotation, when this is actually thrown back in here, these superficial layers passively starts growing inside. So, this passively starts growing inside, but these deep cells which are now moving, so they deep cells starts involuting, and as you can see they start growing beneath this layer of cells which is the blue or the red cells. So, they start from here and now they are growing inside in the inner surface of this particular cells, and therefore this kind of movement is termed as involution. And this is then how later at the end of the mid gastrulation it is formed, that this is the archantron cavity, it is lined by the yellow cell, and this is basically the arrows show the movement. So, the marginal zone cells started moving from here and now they have started growing inside and going up here. There is an opposite direction of basically the blue cells, or which is the ectodermal cell, as they grow by the epibole, which is growing over the surface of other cells and covering it. So, now they will try to expand on both the sides, but these marginal zone cells have started involuting in this particular manner. So, as the new cells enter the embryo, the blastocele is displaced to the side opposite the dorsal lip of blastopore. Meanwhile, the lip expands laterally and ventrally as the process of bottle cell formation and involution continue around the blastopore. So, this is just showing the dorsal lip of blastopore. So, as it keeps on happening, we can see here, as it keeps on happening, so this was happening at this end which is the dorsal, then it starts happening at the lateral sides also, and then it starts happening at the ventral. So, this is basically the ventral side. So, it starts happening at the ventral side also. So, there are the bottle cells which are there, and they will again go and they will start involuting. So, here you can see this entire blastopore is now formed. There is a dorsal lip, there are lateral lips, and there is a ventral lip, and in the ventral side also this thing is the marginal zone cells are involuting and moving towards this side. So, the red is already moving here, the yellow has been lining the gut cavity, and this will also start moving from here in this particular direction, and the anterior endomesoderm has reached up to this point. So, they started from here and now it has reached, the anterior endomesoderm has reached up till this point basically. So, the lip expands laterally and ventrally as the process of bottle cell formation and involution continue around the blastopore. The widening blastopore crescent develops lateral lips and finally a ventral lip over which additional mesodermal and endodermal precursor cells pass. So, the endodermal cells and mesodermal cells, they keep on passing. First they start from the dorsal side, then they go on to the lateral side, and then they go on to the ventral side, so that the entire pore is established, and all the endodermal as well as the mesodermal cells, they start going inside the embryo. With the formation of the ventral lip, the blastopore has formed a ring around the large endodermal cells that remain exposed on the vegetal surface. This remaining patch of endoderm is called the yolk plug, and yolk plug too is eventually internalized. So, you can see in this particular condition that this is the ventral lip now that has formed. So, this was the dorsal lip, these are the lateral lips, and this is the ventral lip that has now formed. And when this ventral lip is formed and the external cells, the endodermal and the mesodermal cells are going inside, then there is large endodermal cells that remain outside, at the later stage or the last stage. This forms a kind of a plug in the hole, and this is termed as the yolk plug. And this yolk plug is, so you can again see the kind of movements that is occurring. So, the movement is basically the cells are growing inside and then from here it is going inside and then moving in this particular direction, and this yellow portion is the yolk plug, but eventually this yolk plug will also be internalized and there will be no endodermal cells which are appearing on the outside. An important aspect is talking about that how this dorsal mesoderm, going into a little detail of how this dorsal mesoderm is forming. So, we have followed how the endoderm is taken inside, and now we will follow how this dorsal mesoderm is basically going to be formed, and there is a phenomena which is termed as convergent extension. So, involuting marginal zone is originally several layers thick. So, involuting marginal zone is the marginal zone cells which is differentiated into the cauda mesoderm, which is going to give rise to the notochord and the somites, and the head mesoderm, which is going to give rise to the head region. So, the head mesoderm and the cauda mesoderm are the two mesoderm which are lying in the mesoderm in the marginal zone region, and that has to be involuted. But initially they are several layers thick. Shortly before their involution, several layers of deep IMZ cells intercalate radially to form one. So, when we talked about general movements of gastrulation, we talked about two kinds of intercalations. One is called as the radial intercalation and another is called as the mediolateral intercalation. So, radial intercalation is when multiple layer of cells become incorporated, become integrated into one another and they became a single layer. So, involuting marginal zone to start with is many layer thick, but then these layers they get arranged and they become a single layer. So, such kind of intercalation is termed as radially. So, IMZ is originally several layers thick, but shortly before their involution, several layers of deep IMZ cells intercalate radially to form one thin broad layer. This intercalation further extends the IMZ vegetally. So, vegetally this intercalation is basically going to expand. At the same time, the superficial cells spread out. So, this is the involuting marginal zone, which are basically we are talking about the deep cells, and at the same time the superficial cells they spread out by dividing and flattening. When the deep cells reach the blastopore lip, they involute into the embryo and initiate a second type of intercalation which is called as a radial type of intercalation. So, what happens is that first the multi-layers become a single layer, and then this single layer will become a narrow, it will form a single longer layer in which the cells now intercalate to give rise to a elongated or involuting kind of a layer. So, you can visualize this radial mediolateral intercalation, it will elongate in mediolateral direction. You can visualize this mediolateral intercalation by the way that when many cars are coming from the highway and they come together and they kind of converge, many different lanes converge onto the highway. So, the cars are coming from different lanes, and when they converge to the highway which is now narrower, they will start coming one beside the other. So, they are kind of they are different cells which are there, and these cells kind of arrange themselves in a mediolateral direction one after the other. So, that leads to the elongation, that leads to the infoliation, or that leads to the movement in the anterior direction which is there. So, at the same time these superficial cells spread out by dividing and flattening. When the deep cells reach the blastopore lip, they involute into the embryo and initiate second type of intercalation which is termed as the mediolateral intercalation. This intercalation causes convergent extension. So, it is convergent because many cells are coming together, and also elongation. So, extension, convergent extension along the mediolateral axis that integrates several mesodermal streams to form a long narrow band. The anterior part of this band migrates towards the animal cap. So, you can visualize in this particular diagram, this is the involuting marginal zone. So, initially this involuting marginal, so these are the deep layer of the involuting marginal zone, this blue is the ectodermal layer. So, this deep layer is several cell layers thick as it is shown here. So, the first thing will be these several layers where they will radially kind of, and they will become a single cell layer, and therefore that will be like how expanded it would be. And this expansion will later on will also cause the elongation, because this first multi-layer will become a single layer, and then single layer will converge to one layer of cells which will be elongated in direction. So, you can see that it is a 3-dimensional structure. This is the bottle cells which are starting. So, they have started invaginating from this point. This is the developing archentron which is there. This is the blastocele that is displaced. And these are the precursors of the pharyngeal endoderm and head mesoderm. Usually the pharyngeal endoderm and head mesoderm, it is difficult to separate them, and therefore they are termed as the endomesoderm, mesodermal region, and then there is a chorda mesodermal region. So, the first one to go inside is the pharyngeal endoderm, followed by the head mesoderm, followed by the chorda mesodermal cells which are there. So, they have reached up to the anterior part, and this anterior part is happening because of this kind of an extension. So, this is basically the radial intercalation, and this is how the mediolateral intercalation is happening, and these they are moving towards the anterior side. So, convergent extension of the dorsal mesoderm is very important. So, now we have seen how the endoderm has been internalized, and how the mesodermal tissue, particularly the dorsal mesodermal tissue, has organized themselves to lie in between the outer surface cells as well as the inner surface cells. Now, outer surface cells, when we looked at the fate membrane, the ectoderm cells were basically on the animal hemisphere, but now they have to grow and expand and cover the entire embryo from the outside, the surface epidermis, and this is done by the gastrulation mechanism which is termed as epiboly. So, epiboly of the prospective ectoderm during gastrulation, the animal cap and the non-involuting marginal zone cells expand by epiboly to cover the entire embryo. These cells will form the surface ectoderm. As the ectoderm spreads over the entire embryo, it internalizes the endoderm within it. The dorsal portion of non-involuting marginal zone extends more rapidly towards the blastopore than the ventral portion, causing the blastopore lips to move towards the ventral side. So, this is, you can see from here that this is the dorsal slit, it was formed, and now this region is actually extending. So, you can see the difference in the pigmentation here. So, this animal cap is highly pigmented, and now they start growing, and now they have start expanding up till this region, and now they have kind of engulfed the entire. So, this is the dorsal lip of the blastopore which is formed, then these are the lateral lip of blastopores that lip appeared, and this is the ventral lip. So, when dorsal lip and ventral lip has appeared, it kind of formed a ring, and now it is surrounded by all the endodermal, the ectodermal cell or the surface ectodermal cells which are there, and all the endodermal and mesodermal cells have been internalized through this pore. Now, as the dorsal surface is growing more, it is more towards the ventral side, and this later on becomes anus. So, it becomes really small, and so this is how this happened. So, while those mesoderm entering through the dorsal lip of the blastopore give rise to the central dorsal mesoderm, the notochord and the somites, the remaining of the body mesoderm enters through the ventral and lateral blastopore lips to create the mesodermal mantle. So, the mesoderm which enters through the dorsal side usually give rise to the notochord and the somites, and that is why we said it is called as the chorda mesoderm, and rest of the body mesoderm enters through the ventral or the lateral blastopore lips. The endoderm is derived from the superficial cells of the involuting marginal zone that form the lining of the archentron roof, and from the subblastoporal vegetal cells that become the archentron floor. So, we have already talked about this, that endoderm is internalized, and now they are derived from the superficial cells of the marginal zone and also the vegetal endodermal cells which are basically forming the roof of the floor of the archentron. So, this gut cavity is lined on both sides by the endoderm. The remnant of the blastopore where the endoderm meets the ectoderm then forms the anus. So, at the end of the gastrulation, all the endodermal precursors have been brought into the interior of the embryo, the ectoderm has encircled the surface, and the mesoderm has been brought in between. So, this is how the end gastrulation part is, that basically now the blue portion you can see the animal pole where it has come because it has done by epiboly. So, animal hemisphere cells now have surrounded, these are the ectodermal cells which have been surrounded. Now, from the dorsal side, this mesoderm, the red portion which has gone inside, this is going to give rise to the notochord and the chord, and so this is the dorsal blastoporal lip. and this is the ventral blastoporal lip, which ventral mesoderm, and this is the ventral blastopore lip which is there. So, you can see this is the gut cavity or the archantron cavity, and this cavity is lined on both sides by the endodermal cells which have again been internalized, and these yellow cells are the endodermal cells. So, the gut cavity is formed and the 3 germ layers, and these cells, the mesenchyme cells, are mostly on the lateral lip, and the ventral mesoderm give rise to the different body mesoderms rather than the cauda mesoderm which is there. So, to summarize, gastrulation in frog embryo is initiated on the future dorsal side excuse me, dorsal side of the embryo, just below the equator in the region of the gray crescent. The cells invaginate to form a slit-like blastopore. The main body of each cell is displaced towards the inside of the embryo, while the cell remains contact with the outside surface by a way of a slender neck. These special cells are called as the bottle cells, and they line the primitive gut or the archantron as it's formed. The next phase of gastrulation involves the involution of the marginal zone cells, while the animal cells undergo epiboly and converge at the blastopere. The first cells to compose the dorsal blastopore lips are the bottle cells. They later become the pharyngeal cells of the foregut. So, pharyngeal endodermal cells, basically they are passively moving and they are the first ones to form. And as I said, the composition of the cells at the blastomere, particularly the dorsal lip of blastopore, keep on changing. So, at first it is basically the pharyngeal endodermal cells, then there will be the head mesodermal cells, then there will be the cauda mesodermal cells, and so the cell composition keeps on changing. Then the dorsal blastopore lip become composed of cells that involute into the embryo to become the precaudal plate, the precursors of what is called as the head mesoderm. So, the pharyngeal endoderm will be followed by the head mesoderm, and the next cells involuting into the embryo are called as the cauda mesodermal cells. They form the notochord, a transient mesodermal backbone that plays an important role in distinguishing and patterning the nervous system. So, the pharyngeal endoderm followed by the head mesoderm followed by the cauda mesoderm. The pharyngeal endoderm and all these 3 cells that start involuting is from the marginal zone and from the dorsal blastopore lip, that is the first structure for the gastrulation to begin that has appeared. So, everything is moving from the dorsal blastopore lip, pharyngeal endoderm followed by the head mesoderm followed by the cauda mesodermal cells. Now, as the new cells enter the embryo, the blastocele is displaced to the side opposite to the dorsal lip of the blastopore, and the blastopore lip then expands laterally and ventrally as the process of bottle cell formation and involution continue about the blastopore, and then eventually the entire ring-like blastopore is formed, and all the endodermal cells, they have been internalized. The mesodermal cells have been placed in between the ectodermal cells, and the ectodermal cells grow by epiboly. So, with the formation of the ventral lip, the blastopore has formed a ring around the large endodermal cells that remain exposed on the vegetal surface, or it is termed as the yolk plug, that is also eventually internalized, and in the later stage this blastopore, which is towards the ventral side, becomes the anus, and the 3-layered body plan of the embryo is being achieved. Thereafter comes the process of induction and the process of neurulation on the formation of the nervous system, and we will follow that in case of amphibians in a separate session. Thank you.
Thank you, ma'am. Thank you so very much. Dear friends, we would like to tell you all that all the lectures pertaining to the series on development biology are already there for you on YouTube. This lecture is also going to be uploaded very soon. So, watch these lectures, and afterwards if you feel that any query you would like to be answered, then you can contact us. Our email ID is info.cc@nic.in. We would be meeting again soon. Till then, take care. Goodbye. Thank you, ma'am. Thank you so very much.